Greetings, everyone. We are Team Eureka 7 from Guru Gobind Singh Indraprasth University. This is our submission for Brainwave 2026 under the Open Innovation Track. Our project is a 3D brain tumor segmentation system, an AI that can look at a brain MRI and automatically outline a tumor the way a radiologist would. But instead of hours, it would do it in seconds. So let's get into it. Let's start with the problem that we are solving. When a patient gets an MRI scan for the suspected brain tumor, someone has to look at the scan and mark out exactly where the tumor is, the shape, the boundary, the texture, where the tumor is swollen, where the which part of the tumor are active tumor. Today, that job is done manually by slice by a radiologist, and a single brain cell can have over 100 slices. To make that concrete, a radiologist often need to go through several different MRI volumes at the same patient's brain just to work out what type of brain tumor they are dealing with. Before predicting how it should be segmented, there's a lot of manual cross-referencing for a single diagnosis. So the real question we asked was, can an AI system do this segmentation automatically, fast, accurate, and consistent every single time? So the radiologists spend less time tracing and more time deciding on the treatments. Our answer is a brain tumor segmentation system that automates this entire process using two deep learning models working on the same pipeline. So we could actually compare their strengths: 3D unit and a Swin transformer. 3D unit CNN based. 3D unit is a convolutional neural network designed for volumetric medical image segmentation. It uses an encoder decoder architecture with a connection to learn both high level features and fine spatial details from 3D MRI volumes, enabling precise localization and segmentation of brain tumors. Coming to Swin Transformer, based on NVIDIA 2021 research paper, Swin Transformer is a transformer based segmentation model that employs shifted window self attention to capture both local and long range spatial relationship within 3D MRI scans combined with the convolutional decoder. It provides accurate and robust tumor segmentation, particularly for complex tumor structures. Technical approach, this is the heart of the project. So let me walk you through exactly how a scan moves through a system step by step. Step one, data input. A patient scan is just not an image. It's four MRI modalities: T1, T1CE, T2, and flare, plus the ground truth tumor mask used during training. Each modality highlights something different. For example, T1CE lights up the active tumor boundary using contrast enhancement, while flare is best at showing where the swelling is around the tumor. Step two, pre-processing. We load the nifty medical imaging files, normalize the intensity values, so brightness is consistent across scans, resize everything to a standard 128 cubed volume, and remap the raw tumor labels into a clean, contagious, and a set of classes. The result is a uniform 4-channel 3D block of data, the same shape every time, regardless of the original scan. Step three, model training. This standardized data is fed into whichever architecture we are training, and the 3D unit or the training. The 3D unit or Swin transformer, we train on 3D patches rather than the full volume. So it fits into the GPU memory. Using a combined dice plus cross entropy loss with class weighting, because the vast majority of voxels, and we need the model to not ignore the small tumor regions in favor of just predicting background everywhere. Step 4, saved models. Once trained, each model is saved into 3D unit Keras or Swin transformer Keras, so we do not have to retrain from scratch every time we want to run the inference. Step 5, the inference API. This is where it gets clever. A full brain scan is too large to process in one shot. So we use sliding window inference. We scan across the volume and overlapping 128 cube patches, 60 and stride 64, and the average overlapping prediction together before taking the final class with argmax. This means we get a full resolution result without ever exceeding memory limits, and there are no visible seams where patches mean this is served through a backend API. Step 6, a front end UI. Built in Streamlit, a doctor or user uploads their 4 scans, pick which model to run, and gets back a slice by slice visualization with the predicted tumor overlaid directly on the scan, downloadable on the segmentation file. So end-to-end raw scans in a fully clean tumor map, fully automated in a fraction of a time manual tracing would take. So here's what actually feels like for a user. In plain terms, you upload 4 MRI volumes on our website. They travel through fast API to our server where pre-processing happens automatically. Then pick which model you want to run, 3D unit or Swin transformer. That model does the work and the segmentation comes straight back to you through the same API for you to download. You can view the report directly on the website, see exactly where the tumor is on each volume, and download everything for the rare cases where our model does not capture every detail perfectly. A doctor still have to go and make some small manual corrections. This is built to assist radiologist judgment, not to replace it. If we go through the if we go through our website, you can clearly see here you can upload all 4 MRI modalities, and you can run the segmentation. It will take a while in our server to do the segmentation. You can see our website have detected the tumor in the medical images that we have provided. Here, different type of colors states to the different type of tumors. And now you can download the segmentation from here. That's it. It's very easily accessible and useful. Coming to the feasibility and viability, our project is completely technically feasible. It uses 3D unit and Swin transformer and built with TensorFlow, Streamlit, and API. It's easy to deploy, scale, and maintain. Viability, if I say, it reduces manual workload, enables faster diagnosis and treatment planning, integrates easily into existing clinical workflows, and has a very strong potential for real healthcare adaptation. If we talk about the features of our project, accurate segmentation, multi-model MRI support, advanced AI models, a fast and efficient inference, visualization and analysis, user-friendly interface, easy data upload, downloadable results, reliable and robust, scalable and extensible. Talking about the tech stack, quickly on what this is built with: Nibable and NumPy for loading and pre-processing the medical imaging data, TensorFlow and Keras with CUDA acceleration for training both architectures. Model saved in Keras format, fast API serving inference with NumPy and Pydantic and Streamlit with Plotly for the visualization layer underneath that Docker, GitHub, Kaggle for datasets, and Monai as a core medical imaging library, all standard, well-supported open source tools. Coming to the impacts and benefits, then the early tumor detection, faster diagnosis, higher accuracy, improved healthcare accessibility, better patient outcomes. And the benefits are that you generate fast and reliable segmentation result. It supports visualization and volumetric tumor analysis. It's very easy to use, built with fast API and Streamlit. Cost effective because we have made it with the uses of open source technologies, ready for integration into AI assisted clinical workflows. So everything you have seen is fully open and reproducible. Our GitHub repo, full documentation in the README, and the demo video walking through the working Streamlit app, all linked here and publicly accessible. To sum up, brain tumor segmentation today is slow, manual, and inconsistent. We built an automated dual model end-to-end pipeline from raw MRI scan to a downloadable 3D tumor map using proven open source AI. It's fast, it's reproducible, it's genuinely deployable, and not just a notebook experiment. That's Eureka 7. Thank you. We are ready to have any questions.